Okay, so that video, we talked a little bit in the video about replication usually results in death and lyse of the host cells. So there are 5 stages. It attaches to the cell. Here's a cell, attaches to the cell. It puts the DNA, right? It attaches and then it like releases the DNA or RNA in there. It synthesizes that DNA or RNA, it assembles it, and then it releases it back out to infect more. So those are the 5 major stages. We watched that video. Here is exactly what happens. So here's the attachment, that it's going to release all of this DNA or RNA, right, into the cell. Okay, so attachment, then release. Here's the DNA. See it right there? Then it's going to synthesize that. Then there's going to be a bunch more DNA. It's going to replicate inside the whole thing and make more little phages. Then it's going to assemble those phage heads and everything all together. Then when everybody's ready to go out into the great big world, it's going to release those and reinfect everything. Okay, so that's the main replication cycle. It does this, like there's an entry period and then the synthesis and assembly as they're getting ready to make more, and then a release of virins by lysis. So this is the time attachment, the burst time, right? This is from the time the phage attaches to the cell. It takes about 25 to 30 minutes before it actually bursts and makes more. Okay, here's another one. This is viral replication. Here you go.
Virulent phages carry out a simple life cycle called the lytic cycle. The phage attaches to the host cell during the attachment stage and then penetrates the host cell and injects its DNA during the penetration stage.
After penetration, the phage causes the host DNA to break into small pieces. The phage then uses the host machinery to synthesize new copies of its DNA. This process is part of the biosynthesis stage. Biosynthesis also involves production of viral proteins. Once biosynthesis is complete, the phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages go on to infect other cells.
Fast forward. Attachment, penetration, there's your DNA. Then it starts replicating itself, breaking apart the regular DNA, biosynthesis. Then it forms the assembly. Once the Avengers are assembled, then they're ready to go out into the world. There you go. That is the lytic cycle of viruses. Okay. So again, you have your attachment. The DNA is going in the entry. It's going to prophase into the chromosome, and then it's going to release into the rest of the world. Very similar in each one of these. Okay. Lysogenic replication. We will actually do that one in a second. Again, prophages have inactive phage stages and results when phages carry genes that alter the phenotype of the bacteria. That is just this part where you go in and it just actually changes the DNA inside the host cell to do something else, to grow a tumor. Typically, this is what you have when you have a viral infection that causes you to grow some kind of tumor or changes your actual cellular structure. Location, I don't know if I have this one. Hold on, let me look. Viral, here we go.
Temperate bacteriophages carry out 2 types of life cycle, the lytic cycle and the lysogenic cycle. The lytic cycle for temperate bacteriophages is similar to the lytic cycle for virulent phages. The phage attaches to the host cell during the attachment stage and then penetrates the host cell and injects its DNA during the penetration stage. In temperate bacteriophages, the phage DNA forms a circle, which can either replicate and be transcribed to produce phage components in the lytic cycle, or can proceed to the lysogenic cycle. During the biosynthesis stage of the lytic cycle, the phage DNA directs the host cell to synthesize viral components. The phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages can go on to infect other cells.
Right, that's just like the cycle that we already talked about. The lysogenic cycle. Boom, boom, boom.
Recall that temperate phages can proceed either to the lytic cycle or the lysogenic cycle shortly after penetration. In the lysogenic cycle, the phage DNA integrates within the bacterial chromosome by recombination. The inserted phage DNA is called a prophage. Most of the phage proteins are repressed by two repressor proteins that are products of the phage genes. Whenever the bacterium reproduces, the prophage is also copied. The prophage is excised from the host chromosome in a process called induction. Induction can occur spontaneously through recombination or some other genetic event, or through the action of UV light or certain chemicals. At this point, the phage may enter the lytic cycle.
Okay, so two types. This one is going to just release out into the world. This one is going to actually combine the DNA into the genome. Okay? The idea is it's going to cause tumor growth, or it's going to cause some kind of genetic change. Okay? All right. Replication of animal viruses, same replication pathway as bacteriophages. There's a presence of an envelope. They don't have cell walls, so the process looks almost exactly the same, except there's a chemical attraction between the viral protein and the cell receptor. They don't have tails or tail viruses. There's direct penetration and endocytosis, which looks like this. So you have your viral DNA right here. It's going to just kind of set on top of this because there's no cell wall, remember, it's an animal cell. And it's going to, again, just inject that viral genome right directly into the cell. In some cases, it's going to endocytose, which means the cell looks like this, and it kind of just comes up, grows around it, and just takes it in. That's called endocytosis. It just kind of grows around it until it's enveloped in there. And membrane fusion looks almost exactly like endocytosis. It just fuses with the membrane and then injects. All three of these, same thing. It's just going to inject the DNA into the host. Here it is again, all three of them, single-stranded, double-stranded, doesn't matter. It's all going to get put into the host and replicated into the DNA. Retroviruses do not use their genomes as mRNA. They use DNA, and they use a DNA intermediary, and then it's actually transcribed into the RNA. Very interesting. So this is some of those, how the single-stranded DNA comes in and then it gets transcribed. Here's some examples of that. Most DNA viruses assemble in the nucleus. Most RNA viruses developed in the cytoplasm. I'm not going to ask you any of these specifically. I'm just telling you so that you know.
Viruses are agents capable of infecting cells and reproducing, but it can be argued whether a virus is alive
or just a complex group of molecules, since it requires a host cell in order to reproduce. Whether they are alive or not, they're able to co-opt the host cell's machinery in order to do the bidding of the virus, the replication of the genetic material of the virus, as well as the structural elements that package and protect the genetic materials and enable the virus to infect new host cells. There are many different types of viruses infecting a large range of host cells. They all follow the same general life cycle with variations in the details of how each accomplishes its replication. Viruses are agents capable of infecting cells and reproducing, but it can be argued whether a virus is alive or just a complex group of molecules, since it requires a host cell in order to reproduce. Whether they are alive